Welcome to the motor system and the control of action. This picture is designed to give you a sense of where motor neurons are in the spinal cord. So here you have a beautiful immunofluorescent staining of the spinal cord, cross-section through the spinal cord. You have the dorsal horn up here, so this is dorsal. If you remember, this is top, like the dorsal fin. You have the ventral horn here. This is ventral, meaning belly. And right here in the ventral horn is where the large alpha motor neurons live. And these are closer up stains, immunofluorescent stains of the motor neurons themselves. These motor neurons are really large. They can get up to 40 microns in diameter. These stains are for two different serotonin receptors. But mostly I just wanted to orient you to the spinal cord. So this is all gray matter in here. Again, this is where the motor neurons reside in the ventral horn. These are white matter tracts going to the periphery and up to the brain. I want to clarify a little bit of terminology so that you don't get confused between the motor pool and the motor unit. And no, the motor pool is not something that picks you up to take you to the airport. So the motor pool is comprised of all the motor neurons that innervate one muscle and are clustered together. So here you're looking at these neurons are clustered together and they innervate one muscle fiber. All of the motor neurons that innervate one muscle are clustered together. So here's the ventral horn here. This is just an enlargement of this area here, and you see the motor neurons that would subserve the axial muscles. They're going to be right around here. Extensors, those motor neurons will be here. Distal muscles, those are muscles that are further away. Those will be out here. So the motor pools would be numbers of motor neurons here that innervate specific muscle fibers. And here they show you a mixed spinal nerve. So let me tell you what comprises a mixed spinal nerve. You have axons that leave the ventral horn, these large motor neurons here, and they go out to the periphery to innervate muscles. And they can go, as far down as your big toe to innervate the big toe so that the big toe can move. So you have long stretches of axons here going out to the periphery. You also have here your dorsal root ganglion. So that means that you must have cell bodies here that are sensory neurons that are receiving sensory information through this nerve to the cell body and then synapsing on neurons in the ventral horn, the dorsal horn, of the spinal cord. So these sensory fibers synapse on neurons in the dorsal horn, which then send projections up into higher cortical structures, for example, to the thalamus and other subcortical areas. So a motor pool is all of the neurons here that then subserve a specific muscle. Then the motor unit is one motor neuron that innervates several muscle fibers. So it's a motor neuron and all of the muscle fibers that it innervates. That's what forms the motor unit. An action potential in a motor neuron causes all the muscle fibers it innervates to contract. So here's one motor neuron that's touching three different fibers, and one action potential here will cause all three of these fibers to contract. Every muscle in the body is composed of many motor units, but the number of fibers in a motor unit varies. So there's a small number of fibers in motor units that exert fine control, such as eye muscles, whereas large motor units are used for powerful movement. So, in vision then, if you didn't have that distinction, you had large motor units subserving the fingers. You tap someone on the shoulder and no longer a fine movement. This is a gross, heavy movement that then bowls them right over. So you need to have distinctions between fine control and gross control. So muscle force can be graded. So graded is just a series of gradations, a series of incremental changes by changing the number of motor units recruited by higher centers. So as you might expect, muscles that have a large number of small motor units are capable of fine gradation of force. So just a fine incremental increase, a small incremental increase. and that's by having a large number of small motor units, whereas some muscles have a small number of large motor units and are perfect for brute force. So again, the basic principle is a motor pool consists of many motor neurons, so here they are, each of which innervates a motor unit within the muscle. So the motor unit is this one cell innervating several fibers. And this slide should be pretty reminiscent of what we discussed with action potentials. There are a lot of the same kind of features or patterns here. So a motor unit contracts in an all or none manner. And remember, that's like the action potential is all or none. However, muscle tension can be varied by changing the number of motor units responding at one time. So it's recruiting more motor units, changing the frequency of motor neuron action potentials, that's rate coding, and we've seen that before. So when you increase the stimulus, you don't increase the action potential, you increase the number of action potentials, the amplitude being the same. So kind of the same principle here by changing the frequency of motor neuron action potentials, and also by changing the type of motor unit that is active. One thing to remember here is that the muscle fibers within a motor unit are homogeneous, meaning the muscle fibers within a unit are all the same. Muscle fibers vary in terms of contractile speed and degree of fatigue, so the fibers will vary, but the fibers within a unit are the same, so that doesn't vary. So then, again, grading is a series of successive changes, incremental changes, successive stages, systematic progression. So remember that. So one way to grade force, that is to get systematic progression in a force, is to vary the frequency of motor neuron action potential firing. And again, this is not a new concept to you. So this is accomplished by increasing the strength and number of synaptic inputs from higher brain centers or from sensory nerves to the motor neuron within the spinal cord. So it's increasing what? The stimulus. And that's the same thing we saw in an action potential. One way to vary the frequency of an action potential is to increase the stimulus. Same thing here. One way to increase the frequency of motor neuron action potential firing is to increase the stimulus. The stimulus comes from higher brain centers or from sensory neurons. As the motor neuron fires more action potentials, the muscle force summates and builds. So it builds together. The more action potentials, the muscle force sums together, it builds. So what are the different motor unit types? You have a slow motor unit, which means low force, a slower contraction speed, but highly fatigue resistant, so it can stay active for a long period of time. So it's slow onset, slow contraction speed, but enduring. Next you have FR, which is a fast fatigue resistant motor unit. So this comes on fast, and yet it maintains its amplitude for some time. Finally, you have a fast fatigable motor unit, which comes on fast, and then within seconds to minutes, it fatigues, it's tired, and becomes quiescent. This slide is simply showing that you can measure alpha motor output to muscles using an electromyogram, EMG. So here you have extension of the elbow. So you have your triceps then that are contracting. So, do that while you're sitting there. Extend your elbow, and it will be your triceps muscle that's contracting. Your bicep muscle is stretching. So you don't see a reaction there. You see it in the triceps here, and you have an extension of the elbow. So when you have a flexion of the elbow, so extension is to move outwards, flexion is to flex, to move inwards, then you have the biceps contracting, and the triceps are rather quiescent. And here you have a classic example of a monosynaptic reflex arc. And I want you to remember this, the monosynaptic reflex arc. So there's no interneuron in this pathway that leads to contraction of the quadriceps muscle. The bipolar sensory neurons synapses directly on a motor neuron. Okay, so you see this here. synapses directly on the motor neuron. And you've seen these sensory receptors already. This is the muscle spindle. So if you remember from the last chapter, this is the stretch receptor. So this is synapsing directly on the alpha motor neuron here. So that when you strike at the knee here, you're striking the patellar ligament. It's just below the patella, the kneecap, and it stretches the muscle spindle and the quadriceps muscle here. This then, the striking here, causes stretch in the stretch receptor, which then carries that message to the alpha motor neuron here, causing the alpha motor neuron to promote contraction of the quadriceps muscle. So again, you have sensory receptor synapsing directly in the spinal cord on the alpha motor neuron that then comes out and tells this muscle to contract. And this, in a sense, this helps us to maintain stability due to an unexpected stimulus. So it helps us maintain, if someone pushes us, we have that same kind of reaction. It allows us to respond immediately, very quickly, to a stimulus without having to send those signals all the way up to the cortex. So all of this monosynaptic reflex happens at the level of the spinal cord without input from descending cortical structures. Now let's look at the different ways that the brain actually innervates the spinal cord. So you have, over here, you have what's called the pyramidal tract. So you have pyramidal neurons up here in your motor cortex. They're beautiful pyramidal neurons that then send their projections down through the white matter tracts, down through the midbrain to the medulla, where at the level of the medullary pyramids, this then crosses over to the other side and synapses on the other side of the spinal cord. So those are your pyramidal tracts. You have several extrapyramidal tracts. You have your rubrospinal. So rubro is for red. So here you have the red nucleus here in the midbrain, and these neurons originate here and send projections down. They cross over immediately and send projections down just lateral to the pyramids and synapse on the spinal cord here. You have your tectospinal. So this occurs at the cell bodies occur at the level of the superior colliculus here. This is the tectum that then, again, kind of immediately cross over, go very midline, and then to the contralateral side. You have your vestibulospinal. You have the vestibular nucleus here that then tracks down, crosses over, and has connections to both sides. And your reticular formation, the reticulospinal, that has cell bodies at this level synapsing on the same side, ipsilateral. The reticulospinal tract is mainly involved in locomotion and maintaining posture. The vestibulospinal, as you well imagine, so the vestibular system is all about balance. So you would imagine then that the vestibulospinal tract is a primary purpose is to support posture and maintain balance of the body and the head. The tectospinal tract here is to mediate postural movements of the head, so where the head is in response to visual and auditory stimuli. So you hear someone calling your name, you turn to that call, that's mediated by your tectospinal tract. The rubrospinal tract terminates in the cervical enlargement here, and so that gives us to understand that it's primarily involved in arm movement, so it can be both fine motor control and large muscles in the arms. And finally, the pyramidal tract, which is the corticospinal tract, so that's cortex, corticospinal, synapses in the spinal cord. This is involved in voluntary movement, control of voluntary movement, and also fine motor skills. And other brain structures, cortical brain structures that are involved, and subcortical that are involved in movement, you've actually seen these before, so this should be somewhat of a review for you. So there are 5 structures in the basal ganglia. Now, when I explained it in the video, it was possible to show 3 of these together, and this would be the caudate, the putamen, which if you're coming from the side of the brain, you're going to hit the putamen first, then the caudate, this is the head of the caudate here, wrapping around to the tail of the caudate, and then just behind the putamen is the globus pallidus 1 and 2. In addition to those three structures, you have the subthalamic nucleus, or nuclei, here, that's subthalamus, so they're below the thalamus, and then you have the substantia nigra in the midbrain. Now, input to the basal ganglia comes into those structures that we referred to in the neuroanatomy section as the striatum, and that comprises the putamen and the caudate. So the caudate and the putamen are the striatum. And that's what receives, those structures are what receives input coming into the basal ganglia. Information leaving the basal ganglia do so by way of the internal globus pallidus and the substantia nigra. And the substantia nigra, the external portion of the globus pallidus and the subthalamic nucleus serve to modulate the information that's coming out of the globus pallidus, the internal portion of the globus pallidus. These structures will become increasingly important to you as you start to look at movement disorders, specifically Parkinson's disease, which is a movement disorder that involves loss of dopaminergic neurons in the substantia nigra here. And we also have Huntington's disease, which is caused by the loss of GABAergic neurons in the striatum, in the caudate and the putamen, another movement disorder. Now we move to cortical regions involved in motor control. And this is these pictures are why I explained to you in the neuroanatomy portion that we'll really have an opportunity in later chapters to kind of parse these, break them down into their individual and more discrete components. So at that point, it was really more important that you learn the grosser structures, and now we'll break it down into finer structures. And here, if you remember, we have the primary motor cortex, which is known as M1 or Brodmann's area 4. And I didn't really, in your book you read about Brodmann's area, and I haven't really covered it. You probably won't see this a lot until higher levels courses. And so if you remember primary motor cortex M1, primary sensory cortex S1, remember the primary visual V1, and you remember what they are, I'm not so concerned that you know their Brodmann numbers. And as I said, the corticospinal tract originates from pyramidal neurons in the cortex. It also originates from neurons in layer 5 of the cortex, sending projections down all the way through the spinal cord, as far down as the lower regions of the spinal cord. Your book says up to 1 meter. You also have some contributions to the corticospinal tract from the sensory cortex and from area 6, which would be the premotor cortex, and the supplementary motor area here. So if I ask you on a test where the fibers originate from in the corticospinal tract, you're going to tell me primarily in the primary motor cortex here in both the pyramidal cell layer and layer 5, but also that we have fibers from the sensory cortex, from the supplementary motor area, and from the premotor cortex that give rise to fibers that travel through the corticospinal tract. Other areas here that are involved in speech, you have Broca's area, and it's very important that you remember this. So Broca's area is involved in the motor control of speech, such that when you have a deficit in that area, your speech is very halting, I remember it as kind of being broken speech for Broca's area, but it's very halting, cumbersome, difficult speech. You also have the frontal eye fields here in area 8, the frontal eye fields that are involved in eye movements, and you have the posterior parietal cortex here that's involved in the planning of movement. So here you are, Broca's area, speech, the frontal eye fields in area 8 involved in the movement of eyes, and you have planning of movement, which is in the posterior parietal cortex. Your book also identifies the posterior cingulate gyrus here. You remember this is part of the limbic system. And apparently the posterior cingulate cortex is involved in externally guided eye movements. For example, if I hold a pen in front of you, that will be, and your ability to follow that will be mediated by the posterior cingulate area here, and also is involved in attentional mechanism. So it makes sense that you're paying attention to me moving that pen and your eyes are following it. So that's the posterior cingulate gyrus here. This illustration depicts the hierarchical organization of movements. So you have the premotor and supplementary motor cortex regions here and the motor cortex. So if premotor feeds into motor cortex, then that sends signals through the brain stem down to the spinal cord, synapsing on neurons in the spinal cord that send signals out to muscles. So remember, these are the alpha motor neurons here that then project out to muscles in the periphery. So you have motor cortex to brainstem to spinal cord, and all of this is modulated then by the basal ganglia and the cerebellum. Now this is an interesting phenomenon which we actually saw in our lab in Florida when we were working with spinal cord injuries with rats. And if you sever a rat's spinal cord so that there's no descending input and there's no sensory input coming in, you can still get movement in the hind limbs. And what this is it's a phenomenon referred to as central pattern generators. What these are is they're neuronal circuits that when activated they can produce rhythmic motor patterns like walking or breathing or flying or swimming in the absence of sensory or descending inputs. Your book postulates that this offers a mechanistic understanding of the hierarchical control of movement such that these central pattern generators are already present in the animal. Okay, so let's say they're in this area here, such that they're already set up and ready to go, such that it just takes a signal, a voluntary signal from the motor cortex then to synapse on these central pattern generators then to get the movement started. But clearly, in the presence of a severed spinal cord, intentional movement or the voluntary control of movement is absent, but you can still get movement in these limbs from these central pattern generators. And these slides are showing what happens, this afferentation. So these monkeys were deafferented, meaning the sensory signals from the periphery no longer reach the brain, so they're not sensing any sensory information here. And because of that, they tend to not use, or they don't use, this limb that has lost sensory afferentation. However, when the dorsal root was severed on the other side, so it was deafferented on the other side so that no sensory signal was getting in, then you see voluntary control of both arms. So sensory information is not necessary. However, the animal prefers to use the limb that is receiving sensory information, and when that's gone, the animal will use both limbs. Now, this is a pretty cool set of experiments done by researchers at Johns Hopkins in the mid-1990s. So they're recording from a single neuron, and in the top experiment, whoops, let me get back to my pointer here. The top experiment, the monkey starts at center and moves outward, depending on the stimulus. So the idea is to determine whether it's force of movement that's controlling this neuron, or direction, or what's mediating this neuron's firing. So here you have center moving outwards, and here you have outwards starting out here and moving towards the center. And so you can see in the first experiment, you're looking at a single neuron, and it's firing most strongly when the movement is toward the monkey. So you see as it's moving away, you see less, and that kind of makes sense, actually, that we're going to have neurons that fire stronger when something is coming toward us. And here, kind of the same idea. You have starting at a distal location, moving medially, moving to the center, And once again, your strongest reaction is to movement towards the animal. These results pretty convincingly demonstrate activity of cells in the motor cortex correspond more directly with movement direction. So again, movement toward, movement toward. So what they found was that neurons in the primary motor cortex are broadly tuned to the direction of hand movement, such that each neuron has a preferred direction of movement for which its firing rate is maximal. So a population vector then is constructed from the firing rates of many neurons, many cortical neurons in that area, and that tends to point in the direction of hand movement, and that's referred to as a population vector. So population coding is how information is coded in a set of neurons, and the summed activity over all of those cells, the preferred direction of all of those cells, is the population vector. So I know for some of you, you're looking at this and your eyes are already rolling in the back of your head, and let me see if I can simplify it a bit. So for each neuron has a vector. A vector is simply a combination of magnitude and direction. And so, for example, you're looking at this one neuron here, and you look at the rate of firing, and you plot the rate of firing as it goes from 0 to 0, 360 degrees, and you see that its highest rate of firing is at 180. So the preferential movement of this neuron is at 180 degrees. Okay, that would be moving that way. Okay, so let's say you take two neurons then, and you have this one that we just looked at with a preferential movement at 180 degrees, and you have cell 2 whose preferential movement is at 90 degrees. So you sum those together, and for when you want to look at left at 180 degrees, when you sum these two together, the vector moves this way. And then when you want to look at 90 degrees, you sum these together, and the population vector, so this is the sum of these two vectors, the population vector points a little less than 90 degrees. And so here you have a whole bunch of neurons. I think your book says 241 neurons, and you're summing population vectors for each direction. And so for this direction, you see the population vector is here. For this direction, population vector is here. And your book says then for the summed activity, the direction of movement is going to be with the population vector. So you can anticipate, you can predict simply by summing all of these different individual vectors. You sum these as a population vector, and you get the direction of movement. Now the book has a number of other experiments that kind of reiterate this idea, but this is where it gets kind of neat and where you find applications. So they trained this animal, this rat or mouse, to press a lever to control this robot arm that then produces water for the rat. While they're doing this, while the rat is doing this, they're recording from the primary motor cortex, and they're looking at population vectors that arise when the rat is pressing the lever and getting his water. And so what they're eventually able to do was based on the vectors that are signaling from the primary motor cortex. They're using those signals then that have already been identified as movements to actually move the robot arm. So all the rats had to do was think about getting water, think about moving that lever, and that vector would be activated, and that's been programmed to know that once that vector is activated, that lever needs to go this way or be pressed that way, and the rat is able to get water. This led to a really neat experiment by Andrew Schwartz from the University of Pittsburgh, in which they trained monkeys to use a joystick to control a robotic arm, all the while recording from electrodes in the monkey's brain that responded to movement and direction, and then these were recorded, and the monkey's arm was tied back so he couldn't use it, and the monkey then controlled the movement of this robotic arm through the signals that had been trained in the brain. If you want to learn more about this experiment and how it was done, it was published in the journal Nature, 2008, and you can find it in your university library. And this has led to human experiments. Here you look at an electrode array that's to be implanted in the brain, and here's the electrode array, sorry, and you see the size of it relative to a penny, and here it is in the brain, visualized by MRI, and this is then the subject who's using this electrode to move the cursor on a computer. So these brain-machine interfaces really have become much more sophisticated, all because of this understanding of how directions are encoded in neurons in the brain. Here you see the firing of one cell when different directions, when the subject was told to think about moving in different directions, and you see here that movement to the right was preferred for this cell. And different patterns of neuronal firing were identified when preference was for left or for up or for down, and all of these then were used to train the brain-machine interface to move in those directions when that pattern of firing was identified. So what to understand out of all of this? Okay, so specific neurons will fire in response to specific directions, movement in specific directions. When you have a population of neurons, they will fire a pattern in response to a given direction, and that pattern can be encoded into a computer, so that computer can be taught that pattern means turning left or moving right, and consequently when that pattern is again exposed to the computer or to the brain-machine interface, then that signal is identified as that given movement, and the machine will move in that direction. Now this has been a lot of information, some of it a little dense. So we'll stop here, and then the next video on action and motor control will be on looking at deficits or disorders that involve movement control.